When a plant gets sick, it can't just tell you what's wrong. So, how do scientists figure it out? How do they unmask these invisible attackers? Well, today we're jumping right into the world of plant pathology, where the clues are microscopic, and believe me, the stakes couldn't be higher. We're talking about our global food supply. Okay, so our investigation starts right here, at the scene of the crime. The victim? A potato plant. And it is clearly in trouble. I mean, look at it. The leaves are wilting, they're browning, they're dying. But the thing is, the attacker is totally invisible. And that is the mystery we are here to solve. So the big question is, what is causing all this devastation? To get to the bottom of it, a plant pathologist has to think exactly like a detective. They have to follow this trail of evidence that starts out here in the field and ends up under a microscope. Right, so the very first phase of any investigation is to secure the crime scene, right? You gather all that initial evidence. And in our case, that means a really thorough examination of the plant and its surroundings. First things first, it's all about gathering intelligence. What kind of plant are we even looking at? What are the growing conditions like? The location, the irrigation? Then you get to the visual evidence. The detectives, they look at every single symptom, from the very first signs all the way to the most advanced stages of decay. Oh, and a classic field test, you cut a stem and stick it in water. If you see this cloudy stream of bacteria oozing out, well, that's a huge telltale sign of a massive internal infection. Now, what's so interesting here is just how different the symptoms can be. These are the first visual clues. On the left, you've got a pepper that's literally just dissolving into this mushy, soft rot. And on the right, that's a branch oozing a thick gummy substance we call gummosis. Two totally different cries for help from two different plants. And the clues just keep on coming. The corn leaf on the left has these really distinct stripes running all the way down its length. Meanwhile, the leaf on the right looks like it was scalded with hot water. Every one of these symptoms is another piece of the puzzle, and each one points the investigator toward a very specific group of suspects. So, with all these clues gathered from the field, the investigation moves from the crime scene into the lab. The goal now is to actually capture the culprit, to isolate it from all the other microorganisms that are just hanging out on the plant. Getting a pure sample is absolutely everything. Scientists will take a tiny piece of tissue from the very edge of the infection. You can think of it as the front line of the bacterial invasion. They'll sterilize the surface to get rid of any microbial bystanders and then place it in a petri dish. And this step is so critical because a lot of harmless bacteria actually grow way faster than the pathogen. If they contaminate the sample, the real culprit could just get a let. And here we go. This is the result. After a few days, you get these beautiful, distinct colonies growing on the plate. Every single one of those dots started as a single bacterium. We have officially captured our suspect. And now, the interrogation can begin. All right, the first phase of interrogation. This involves some really fundamental tests to get a basic profile of our suspect. This is what we call the microscopic lineup. Okay, so the very first test is the famous Gram stain, and it basically divides all bacteria into two major groups. It really all comes down to their cell wall. Think of it like this. Gram-positive bacteria have this thick, absorbent wall that just soaks up a purple dye and holds on tight. But gram-negative bacteria, they have a much thinner wall, so that purple dye just washes right off, and they end up getting colored pink by a second stain. And this picture right here shows you exactly why that works. Look at the gram-positive cell up top. That thick, absorbent layer is called peptidoglycan. That's what traps the purple dye. Now look at the gram-negative cell below it. See that thin little layer sandwiched between two membranes? It just can't hold on to the stain. It seems simple, but this difference is a massive clue for our detectives. But hey, what if you need an answer faster? Well, there's the KOH test. It's a really quick and simple way to get the same basic information. Is our suspect gram-negative or gram-positive? And the results are just so obvious. You can see them right away. On the left, a gram-positive bacteria mixed with potassium hydroxide, or KOH, it just makes a milky, watery suspension. But on the right, whoa, look at that. The KOH solution literally rips open the gram-negative cells, releasing their DNA, which creates this long, slimy, mucoid thread. You don't even need a microscope for that clue. Okay, so now that we've got a basic profile of our suspect, the next question is all about its behavior. How does it move? So a lot of bacteria use these things called flagella. They're basically tiny whip-like propellers to get around. And just like the make and model of a getaway car, the number of these flagella and how they're arranged are key features that help scientists identify the suspect. But you have to be careful, because under the microscope, things can be a little deceptive. It's absolutely crucial to tell the difference between true movement and something called Brownian motion. Brownian motion is just the random jiggling of a cell that's being bumped around by water molecules. True motility, on the other hand, is purposeful. You can actually see the bacteria running or tumbling with intent. We've examined the crime scene, we've isolated a suspect, we've checked its basic profile and how it moves. Now it is time for the final phase, cracking its unique biochemical code. This quote really just gets to the heart of it all. You see, unlike bigger organisms, a lot of bacteria can look almost identical under a microscope. Their shape and their size just don't give us enough information. So to get a definitive ID, we have to dig way deeper into their physiology.
This is it, the final stage of the interrogation. We're creating a biochemical fingerprint. Scientists put the bacteria through a whole battery of tests, and they're essentially asking it a series of questions. What do you eat for energy? Do you breathe oxygen? Every species has its own unique metabolic profile, a set of answers that provides an almost certain identification.
So, by putting all of it together, the initial symptoms we saw on the plant, the results of the gram stain, how the bacteria moves, and finally its unique metabolic fingerprint, the scientists can finally put a name to the pathogen. Case solved.
But the work is never really over. This whole process just highlights the incredibly meticulous work that's required to protect our food. And it kind of leaves us with a final thought. With countless bacteria emerging and evolving every single day, what new tools and techniques will the next generation of plant detectives need to stay one step ahead of these microscopic assailants?